Good morning everyone. We are Group 4 and today's session, we will discuss the key categories of biomolecules such as proteins and nucleic acids. In grade 9, you have learned that carbon's bonding characteristics result in the formation of a larger variety of compounds. In this lesson, you will understand more about compounds that are essential to our daily life. These compounds belong to our four main classes of biomolecules, which is carbohydrates, lipids, proteins, and nucleic acid. But today, we will emphasize the proteins and nucleic acids. So before we begin, let us define the objectives of this lesson. Number 1. Recognize the major categories of biomolecules such as protein and nucleic acids. Number 2. Differentiate the biomolecules from each other in terms of their structure and function. Let us now proceed to our topic.
So before we discuss about the structure, uses, and the function of nucleic acid, let's define first what is a nucleic acid. So nucleic acids are molecules that code for hereditary traits by controlling the production of protein. They are a long chain of polymers consisting of simpler unit or monomer. There are two types of nucleic acid: the deoxyribonucleic acid or DNA and the ribonucleic acid or RNA. DNA are found mainly in the cell nuclei that contains genetic information that codes for the sequences of amino acid in proteins, while RNA are found in many places in the cell and carries out the synthesis of protein. As I said earlier, there are classification of a nucleic acid. The other one is DNA. DNA is a double-stranded helix. It stores all the genetic information that an organism needs to develop, function, and reproduce. DNA is the polymer of four nucleotides such as adenine, thymine, cytosine, and guanine. So let's move in RNA. RNA is a single-stranded helix. It converts information that is stored in DNA proteins. RNA is polymer of four nucleotides such as adenine, guanine, cytosine, and uracil. Now, we will discuss the structure of a nucleic acid. Nucleic acid are made up of monomers known as nucleotides. Each nucleotide is made up of three components: nitrogenous base, sugar, and phosphate group. Each nucleic acid contains 4 up 5 possible nitrogen-containing bases, such as adenine, guanine, cytosine, thymine, and uracil. Now that we've learned the structure of it, let's identify the function and uses of nucleic acid. Nucleic acids are the hard drives containing the essential blueprint or source code for making cells. They are the main information-carrying molecules of the cell, and by directing the process of protein synthesis, they determine the inherited characteristics of every living thing. To further understand that, DNA replicates and stores genetic information. It is a blueprint for all genetic information contained within an organism. Then, RNA converts the genetic information contained within DNA to a format used to build proteins and then moves it to the ribosomal protein factories.
Good day everyone, my name is Venus Nora from Rizal and I will be discussing to you the structure, uses, and function of proteins in biomolecules. Well, first, let us know the meaning of proteins. Proteins are large biomolecules and macromolecules that comprise one or more long chains of amino acid residues. Proteins are present in all living organisms and include many essential biological compounds such as enzymes, hormones, and antibodies. Based on the molecular shape, proteins can be classified into two types: fibrous proteins, when the polypeptide chains run parallel and are held together by hydrogen and disulfide bonds, then the fiber-like structure is formed. Such proteins are generally insoluble in water. These are water-insoluble proteins. Example, keratin present in hair, wool, and silk, and myosin present in muscles, etc. Number 2, globular proteins. This structure results when the chains of polypeptides coil around to give a spherical shape. These are usually soluble in water. For example, insulin and albumins are common examples of globular proteins. There are 4 levels of protein structure. Number 1, the primary structure of protein is the hierarchy's basic level and is the particular linear sequence of amino acids comprising one polypeptide chain. Number 2, secondary structure is the next level up from the primary structure and is the regular folding of regions into specific structural patterns within one polypeptide chain. Hydrogen bonds between the carbonyl oxygen and the peptide bond amide hydrogen are normally held together by secondary structures. Number 3, tertiary structure is the next level up from the secondary structure and is the particular three-dimensional arrangement of all the amino acids in a single polypeptide chain. This structure is usually conformational native and active and is held together by multiple non-covalent interactions. Number 4, quaternary structure is the next step up between two or more polypeptide chains from the tertiary structure and is the specific spatial arrangement in interactions. What makes up protein structure? A protein's primary structure refers to the amino acid sequence in the polypeptide chain. Peptide bonds that are made during the protein biosynthesis process hold the primary structure together. What determines protein structure? In the polypeptide chain, the main structure of a protein relates to the amino acid sequence. The primary structure is bound together by peptide bonds that are made during the phase of protein biosynthesis. The primary structure of a protein is determined by the gene corresponding to the protein. What is the primary structure of a protein? The linear sequence of amino acids within a protein is called the primary structure of the protein. A sequence of just 20 amino acids, each of which has a special side chain, is made up of 8 proteins. The side chains of amino acids are chemically distinct. Okay, so let's move on to the uses of proteins. Proteins themselves are major structural elements of cells. They also serve as transporters, moving nutrients and other molecules in and out of cells, and as enzymes and catalysts for the vast majority of chemical reactions that take place in living organisms. Proteins are large complex molecules that play many critical roles in the body. They do most of the work in cells and are required for the structure, function, and regulation of the body's tissues and organs. The last thing we need to know about protein is the function of it. They are responsible for number 1, building tissues and muscles. Protein is necessary in building and repairing body tissues. Number 2, hormone production. Hormones are chemicals produced by glands in one part of the body that help coordinate activities and communicate with other areas. Number 3, enzymes. Number 4, immune function. Number 5, energy. There are important functions of protein in your body. Number 1, growth and maintenance. Number 2, causes biochemical reactions. Number 3, acts as a messenger. Number 4, provides structure. Number 5, maintains proper pH. Number 6, balances fluids. Number 7, bolsters immune health. Number 8, transports and stores nutrients. And that's all. I hope you learned something from me.
The different general of structure of the different types of proteins and nucleic acid. Nucleic acids are polynucleotides, that is, long chain-like molecule composed of a series of nearly identical building blocks called nucleotides. Usually, nucleic acids are helix-shaped with a double helix for DNA and single helix for RNA. Carbon, hydrogen, oxygen, nitrogen, and phosphorus atoms are arranged strategically into the building blocks of nucleic acids. These building blocks are ribose sugar and then 4 other components called adenine, thymine, guanine, and cytosine. We also sometimes call these last 4 components nitrogen bases. In RNA, there is an additional base called uracil which takes the place of thymine. In the DNA molecule, all of these components are arranged in a shape that resembles a twist ladder. Notice how the sides of the ladder alternate between ribose sugar groups and phosphate groups. Also notice how adenine always pairs with thymine and guanine always pairs with cytosine. The nitrogen bases are connected across the molecule using hydrogen bonds. Adenine and thymine are connected using 2 hydrogen bonds, and guanine and cytosine using 3 bonds. Like proteins, nucleic acid have a primary structure that defined as the sequence of their nucleotides. Unlike proteins, which have 20 different kinds of amino acids, there are only 4 different kinds of nucleotides in nucleic acids. Protein structures are made by condensation of amino acids forming peptide bonds. The sequence of amino acids in a protein is called its primary structure. The secondary structure is determined by the dihedral angles of the peptide bonds, the tertiary structure by the folding of proteins chains in space. Association of folded polypeptide molecules to complex functional proteins results in quaternary structure. Proteins contain carbon, hydrogen, oxygen, nitrogen, and sometimes sulfur as well. These atoms arrange themselves into something called an amino acid, which looks like this. Notice how one of the carbons has an attachment called an R group. These are actually 20 different R groups that could be attached to an amino acid, and they are what makes each amino acid unique. The R group is the portion of the amino acids that can sometimes contain an atom of sulfur. The 20 different varieties serve as the building blocks for proteins. A series of amino acids is strung together using connections called peptide bonds. Every time we attach a new amino acid to the end of the chain, we remove one molecule of water in a process called dehydration synthesis. When we created a long string of amino acids all connected in this way, we call it a polypeptide. When the appropriate number of amino acids have been added, we fold up the polypeptide into a structure known as a protein. The protein will have a specific shape that's based on the task it will perform. Remember that a molecule is not considered to be a protein until it's been folded. The way the protein is coiled up can also make a huge difference in terms of its function. Proteins may take on a primary, secondary, tertiary, or quaternary structure. When we have a number of proteins who beneath working together, we call this a protein complex. So, the question is, what is the difference between the structure of nucleic acid and the structure of a protein? Nucleic acids are larger compounds made of amino acids, while protein are smaller subunits that make up amino acids. Nucleotides link together to form a nucleic acid. Amino acids link together to form a protein.
Here are the following examples of food that contains protein and nucleic acids: nuts, fish, vegetables, mushrooms, yeast, and meat. Despite limited research, opponents claim that food rich in nucleic acids can offer a variety of benefits, from a stronger immune system to improved digestion and quicker muscle recovery. In addition to sourcing them from your diet, your body can make nucleic acids from scratch. In fact, your body typically produces enough nucleic acids to cover 100% of your needs. You may need larger amounts of nucleic acids than your body can produce, especially in times of illness or injury or periods of growth. In these cases, eating food rich in nucleic acids can help make up the difference. Before ending up on your plate, most food were once living, so this food contains at least some nucleic acids. It's important to consider that nucleic acid levels can vary widely among foods.
Excess protein consumed is usually stored as fat, while the surplus of amino acid is excreted. This can lead to weight gain over time, especially if you consume too many calories while trying to increase your protein intake. On the other hand, a lack of protein can make you lose strength, lose your muscle mass, makes it harder to keep your balance, and slows your metabolism. Having more nucleic acid in the body could lead to stronger immune system, improved digestion, and quicker muscle recovery. And if we didn't have enough nucleic acid in our body, air and information wouldn't get to the cell, and if this were to happen, the cell would die.